Alright, so welcome back. This is going to be screencast number 2 for chapter 17, and what we're going to do today is we're going to look at the second step in protein synthesis, and that second step is translation. If you remember from our very first video, we had looked at the process of transcription, and transcription was the creation of that messenger RNA. And so for that messenger RNA, what we're doing is we are taking the code from the DNA and we are transporting that message out into the cytoplasm and using that code to create our protein. Now, if we're going to do that correctly, what we need to do is we need to use another type of RNA, and that RNA is going to be called transfer RNA. Sometimes we abbreviate it tRNA. So each transfer RNA is going to carry a specific amino acid, and it's going to carry this amino acid in this area right here of the transfer RNA, and it's also going to have something called an anticodon. So if you remember during our discussion of messenger RNA, we had talked about something called a codon. So the anticodon is going to be complementary to that codon on that messenger RNA molecule. So again, over here on the right, you're going to notice right down here towards the bottom of this transfer RNA, you're going to see the anticodon. Now, a transfer RNA molecule is going to consist of a single RNA strand, and it's going to be about 80 nucleotides long. Now, that strand is going to look somewhat like a clover leaf, and the reason it's going to look like a clover leaf is because as this strand starts to bend and change its shape, there's going to be some hydrogen bonding that occurs between those nitrogen bases. And because of that, that's going to allow different parts of this transfer RNA to sort of hook up or connect together, and that's going to give it the shape that you see over here on the right. So translation is going to require two steps. Now, the very first of those two steps is going to be making sure that we connect the correct amino acid to the transfer RNA. And if you look over here on the right, you're going to notice we have a special type of enzyme called aminoacyl tRNA synthetase. And again, it has the ASC at the end, so we know that it's an enzyme. And this right here is going to be that enzyme. So what we're trying to do is we're trying to encourage a chemical reaction to happen between the transfer RNA and, again, that correct amino acid. So that enzyme is going to bring in an amino acid, and it's going to bring in, if you look down here towards the middle, a transfer RNA, again the correct transfer RNA, and there's going to be bonding that occurs between that amino acid and that transfer RNA. And once that is done, then of course, because it's an enzyme, it's going to go ahead and release its product, which is the connected tRNA to its amino acid. Now there's going to be a second step in translation that is very important, and that's going to be the correct match between the transfer RNA and the messenger RNA. Now, as we had said in the previous screen, on the transfer RNA you have something called an anticodon. And remember, we have the codon on the messenger RNA. And remember, those are going to be complementary to each other. So what that refers to is, if we have a G on our messenger RNA, then on the anticodon of the tRNA, then it's going to pair with C. Now, if we have an A on our messenger RNA, then again, it's going to complementary base pair, but it's going to pair with a U. Now remember, there's no thymine when you look at RNA. So we're going to replace that thymine with a U. So this is what they refer to in terms of complementary base pairing between the anticodon and the codon. Now, occasionally you will find some flexible pairing at the third base of a codon on that messenger RNA. And this is going to be considered the wobble area of that codon. Now, the reason this is there is because it actually allows some transfer RNAs to bind to more than one codon. And so down here towards the bottom, you're going to notice that we have two transfer RNAs. Both of them are carrying the amino acid leucine, but if you notice, the normal pairing would be GAG, so we would have CUC, but if we look at that third base pair, there's going to be a little bit of wobbling occurring. And so in this case, we get the G actually pairing with a U. So again, there can be a little bit of flexibility on how that transfer RNA actually bonds with that messenger RNA codon. Now, as we get into our discussion of translation, you're going to notice that, of course, ribosomes have become incredibly important to this process. So ribosomes are going to be there to make sure that we match the transfer RNA codons, anticodons, with the correct messenger RNA codons. Now, the ribosome itself is going to have two subunits. one of those is going to be large and the second is going to be small. And you can see both of these subunits being represented over here on the right. So again, this is going to be our large subunit, this is going to be our small subunit. Now when you think about the structure of a ribosome, it's going to be made up of two things. It's going to be made up of various proteins, and it's going to be made up of a third type of RNA called ribosomal RNA. Now the ribosome itself is going to have three different binding sites, and these three different binding sites are important to the transfer RNA. So again, looking over here on the right, we have one called the E site, we have one called the P site, and there's going to be a third called the A site. Now when we look at the one called the P site, it's really important because what it's going to do is it's actually going to hold on to that transfer RNA that is also holding on to that growing polypeptide chain. And you can see that right down here. Now the A site, on the other hand, is going to hold on to the transfer RNA that has just come in, and that's going to be this site right here, and it's carrying a new amino acid. And what that transfer RNA is doing is it's waiting for the bonding to occur between the growing polypeptide chain and that new amino acid. Then, of course, we have one called the E site, as we had said, and this is going to be considered the exit site. So once that transfer RNA has dropped off its amino acid, it's going to go ahead and be released back into the cytoplasm, and as we said, it has to pick up its specific amino acid in that cytoplasm. So in other words, that transfer RNA can actually be recycled and used over and over and over again. So as we begin building our polypeptide or protein, there's going to be three different stages. One of them is called initiation, the second elongation, and then the third and final step is going to be called termination. Now the initiation stage is going to involve three things. The first one is a small ribosomal subunit, so that small part of the ribosome is going to bind with a messenger RNA strand, and there's going to be a very special initiator transfer RNA. And so down here towards the bottom, again, you can see that small subunit right here, and the messenger RNA is going to be bound to that small subunit, and as we had said, there's going to be a very special initiator transfer RNA that's going to be represented right here. Then what's going to happen is the small subunit is going to move along the messenger RNA, and it's going to do this until it actually reaches the start codon, which is identified with AUG. And it actually has a very special amino acid called methionine. So once it's reached the AUG codon, proteins called initiation factors are going to bring in the large subunit, you can see this over here on the right, that is going to complete what we call the translation initiation complex. And so what that simply means is that the ribosome has been completed, and now we can continue building our polypeptide chain. Now during the second stage of building that polypeptide, remember that's going to be called elongation. And during this stage, amino acids are going to be added one by one to the preceding amino acid. And you can see that over here on the right. So as we do this, each addition is going to involve special proteins called elongation factors. And this whole process is going to occur in three different steps. The first one is going to be the codon recognition. So again, remember our messenger RNA, we read it in groups of threes, and each of those is considered a codon. So we're trying to make sure that that transfer RNA that is coming in is actually recognizing the correct codon so it can come in and deliver that amino acid. Then if you notice, step 2 is going to be the peptide bond formation. So that peptide bond is going to be used to connect the amino acids as they are brought in to continue building that polypeptide chain. And you can see that indicated right here. Then of course the third is going to be something called translocation. And what that simply means is that once that transfer RNA has been used, then we're going to release that used transfer RNA back into the cytoplasm, and again it's going to go ahead and pick up another amino acid with the help of that amino acyl tRNA enzyme, and then of course be recycled and be brought back in if necessary to continue building that protein. Now once we get to the third stage, remember this stage is called termination. And as the word implies, termination basically means to finish up the process. So the A site is going to accept a protein called a releasing factor. Now the releasing factor is going to cause the addition of something different now. It's not going to bring in an amino acid. It will actually cause the addition of a water molecule instead of that amino acid. Now this reaction itself is going to initiate or cause the release of that polypeptide into the cytoplasm. And the translation assembly that is being used will now start to come apart. So again, this releasing factor is really important because if you notice, it's going to bond to a certain area of the messenger RNA that's called the stop codon, which means that we're finally finished building that protein. Now, if you notice over here, after we have that releasing factor present, then we're going to release that final transfer RNA. And then as we had said, we're going to go ahead and release that free polypeptide. And if you notice in the third stage, once the polypeptide has been released, everything that was being used to actually create that protein will now begin to fall apart. Now, what's kind of interesting about building these proteins is that sometimes what the cell can do is it can actually translate a single messenger RNA, but it can do this simultaneously with many, many ribosomes. So what this does is it gives the cell an advantage. It actually enables the cell to make many copies of a polypeptide really quickly. And that's what you see indicated down here. So if you notice all of these little brown units that you see, these are going to be the ribosomes, and each of these are working on that single messenger RNA strand. And so as it's passed through each of these ribosomes are going to be producing the same type of protein or polypeptide. So basically what it does is it helps us to mass produce that particular protein. Now, often translation is not going to be sufficient to make a functional protein. So what that means is sometimes it's going to need to be modified. So just like with transcription, before we sent that messenger RNA out into the cytoplasm, what we had to do is we had to actually do some modifications to it. So in order to actually make that protein functional, there are three things that possibly could happen to that protein. Some of these proteins will actually spontaneously start to coil and fold and actually become three-dimensional in nature. Some of them are going to be activated by very special enzymes, and these enzymes are going to work to cleave or cut up those particular proteins, and there are going to be others that will actually come together to form the subunits of a much more complex protein. So these would be considered modifications that might occur before the protein becomes a functional unit. Now, sometimes during the building of these polypeptides, what we need to do is we need to sort of think about what ribosomes are actually being used to build these proteins. Now, remember when we talked about cells, there are actually two different categories of ribosomes. One of them is considered free, and the second is considered a bound ribosome. Now, those ribosomes that are considered free are going to be found in the cytoplasm. And most of those ribosomes will synthesize or make proteins that will function in that cytosol or cytoplasm. Now, if it's a bound ribosome, it's going to be making proteins of the endomembrane system. Now, remember, any of the membranes that we had talked about that make up the various organelles within the cell, oftentimes, in addition to the lipids that are going to be present in that membrane, there were often quite a few different proteins that were there as well. And so a lot of times the bound ribosomes are going to be responsible for making the proteins that you would find in this membrane. And in addition to making these proteins, bound ribosomes will also make proteins that possibly could be secreted from the cell. Now, ribosomes themselves are actually identical, and they actually can switch from the free state to the bound state and, of course, back again. Now, polypeptide synthesis will always begin and end in the cytoplasm unless, again, this is going to be an exception to the rule, the polypeptide signals the ribosome to actually attach to the ER. So polypeptides destined for the ER or, again, for secretion are going to be marked by a very special signal peptide. And you can see that signal peptide located right down here on this ribosome. Now again, this is going to be a free ribosome currently in the cytoplasm. Now this signal peptide is important because this is going to be actually the indicator to a signal recognition particle, sometimes abbreviated SRP, which is going to allow it to bind to that signal peptide. And when it does that, it's going to bring the signal peptide and its ribosome actually to the ER. So again, if you look down here towards the bottom, this is our ribosome in the cytoplasm. You can see our messenger RNA being used to build our polypeptide. This signal peptide is going to be part of that polypeptide, and this SRP is going to recognize that signal peptide on the ribosome. And if you notice, because of that, it's going to indicate that it needs to basically be sort of implanted or attached to that ER, because we actually have SRP receptor proteins on the membrane system of that ER. And so once it recognizes that signal peptide, then eventually it's going to attach to the ER, and when it's no longer being used, that signal peptide is going to be removed, and then eventually, once translation is done, it's going to go ahead and release the finished polypeptide or protein into the rough ER membrane. Now that we've talked about both transcription and translation, what we need to do is we need to understand that sometimes there are mutation or changes in that DNA. And when that happens, this can actually affect protein structure and the way that that protein actually functions. Now, when we talk about changes in the DNA and how it affects protein synthesis, we call these mutations point mutations. Now, they're called point mutations because actually they result in just one base pair of a gene that's going to be changed. Now, the unfortunate thing is, it sounds like it's not too big of a deal, but even one change in a base pair of a gene can actually lead to an abnormal protein being produced. Now, if you look down here, this is an example of what could possibly happen to hemoglobin with that single base change. So if you notice, this is considered the wild type hemoglobin. So this is our DNA strand. This is the correct code for the normal hemoglobin molecule. And this would be the messenger RNA that would be coded from this strand. Now, if you notice, this is going to produce a normal amino acid within this hemoglobin called glutamine. But if we have a mutation occur within that DNA strand where we have a change in that base, so instead of being T, now it's been changed to A, you're going to notice that now we change our messenger RNA. When we change our messenger RNA, the codon that's represented here that would eventually code for a certain amino acid, again with the help of the transfer RNA, now it's going to change to valine. And when you see this change, a person with this mutation no longer has normal hemoglobin. This person now has sickle cell hemoglobin or sickle cell anemia. Now, when you talk about point mutations, what we do is we actually place them into two general categories. The first one is base pair substitutions. Now, this is a relatively simple type of mutation where it actually replaces one nucleotide with another. So it's basically you're switching that nucleotide out. Now, sometimes with this particular type of mutation, we call them silent mutations because sometimes even if the change occurs, it actually has no effect on the amino acid being produced by a codon. Now remember, this could be possible because of the redundancy in the genetic code. Remember that redundancy basically means that you have more than one triplet or codon that could actually code for an amino acid. Now when we talk about base pair substitution mutations, we oftentimes will categorize them as either being missense mutations or nonsense mutations. If it's a missense mutation, it's still going to code for amino acid, but as we had said, it probably will not code for the right amino acid. But if you get a change that's pretty severe where it actually codes for a stop codon, that's considered a nonsense mutation, because essentially what that does is it leads to a non-functional protein. You're almost guaranteed of this, because once you hit that stop codon, you can no longer continue producing the amino acids necessary to produce that protein. So over here on the right, this is just an example of some very simple base pair substitutions. So this would be considered the wild type. So this is what should happen based on the messenger RNA that you see here. So if you look at our first example of a base pair substitution, this is going to illustrate basically one that actually has no effect on the amino acid sequence. So instead of having the normal C, we are going to replace it with a U. And because of that, if you notice, we had the arrangement of methionine, lysine, phenylalanine, and glycine in the original wild type of this particular protein. And if you notice, even though we had the substitution of the U instead of the C, we still had all four of the same amino acids being produced. So basically you had no change in the protein that was being made. So this sort of demonstrates that redundancy nature in some mutations. But again, if it's a missense mutation and you replace the G with an A, if you notice, that will alter the amino acids that are actually being produced. So we still have methionine, we still have lysine, we still have phenylalanine, but instead of glycine, now what we have is we actually have serine. And so that's going to change the type of protein that we're creating. Now, because we have an amino acid, again, it is categorized as a missense mutation. Now, a nonsense mutation, like you see down here, what we did is instead of an A, we go ahead and replace it with a U. So again, that's where the mutation occurred. We start off with methionine, but it's nonsense because we end up actually creating a stop codon, which is going to lead to a non-functional protein because we can no longer continue adding amino acids to the messenger RNA strand. Now, sometimes we have point mutations where we have an insertion or a deletion. Now, these are considered either additions or losses of nucleotides. So it's no longer a simple substitution. We're either putting something else in or we're taking something away. Now, when you do this, this can have a pretty disastrous effect on the resulting protein. Because what this does is it actually changes the reading frame of the messenger RNA. And we give it a special name. We call this a frame shift mutation because what it does is it essentially shifts the reading frame of your messenger RNA either to the left or to the right. Now, if you look over here on the right, again, we're still using that wild type, again, messenger RNA strand. So wild type would indicate that it's normal. We still have methionine, lysine, phenylalanine, and glycine, just like we had on the previous screen. So this is the normal representation of these amino acids. So if we have a nucleotide that's actually been pulled out of that messenger RNA strand, what you're going to notice is we still have methionine as we get started. We still have lysine. But once we get to the point where we have the missing nucleotide, everything is going to be shifted to the left. And because of that, we're going to read that particular code in a new way. So there's going to be a new triplet to read. And of course, when we read that code in a new way, it's going to produce a new amino acid. So pretty much everything after that, the amino acids most likely will be different. And of course, this is going to cause a pretty drastic change in the protein that we're attempting to produce. Now, this is considered, again, a missense type of mutation simply because we're still getting amino acids being brought in and used to build that protein. Now, if again we have, say for example, an extra U that is placed into that messenger RNA, in other words, it shouldn't be there, but it's been placed here, then this would be considered a nonsense in this case, simply because again we start with methionine, but again we shifted our reading frame to the right. We had to read those triplets in a different way, but we ran against a stop codon. So that's why we call it a nonsense type of mutation. Now, sometimes you can actually get more than one nucleotide either being added or removed. So you can actually have them in groups. And so down here towards the bottom, this would be an example of actually taking away 3 particular nucleotides at one time. So you're actually removing an entire codon. And so if you notice, no frame shift actually occurs, but what we end up doing is we end up taking out one of those amino acids. And it definitely changes what type of protein will be produced at the very end. So again, when you have these mutations occurring, sometimes these mutations just spontaneously happen. And they can occur during DNA replication, they could occur during recombination, or they could even occur during the repair of that DNA. Now, for the ones that aren't necessarily spontaneous, sometimes in the environment there are things out there that could actually be causing these mutations. And we identify these things as mutagens, and they can be physical in nature or they could be chemical. And so if you look at the middle of the screen, this is going to be 3 different categories of mutagens. One of them is going to be radiation. And so there's different ways that our bodies can be exposed to radiation. One of them we don't have a lot of control over, and that would be UV radiation from the sun. Another one is going to be X-rays. And so say for example you happen to be an athlete, if you're exposed to multiple X-rays over your lifetime, that could have an effect on the mutations that might occur within your DNA. Now, some of them could be chemical, and carcinogens are one of those, and cigarettes would be a really good example of a carcinogen. There are chemicals within that cigarette that could actually affect your DNA and cause those mutations to occur. There's been lots and lots of talk of processed foods and preservatives having various types of chemicals within them that could actually cause mutations. And then, of course, subjecting yourself to various cosmetics and cleaning products and any other chemicals for that matter could again cause mutations in your DNA. And then we have a third category called infectious agents, which essentially would be viruses and bacteria. All right, so that's going to finish up our second and final screencast for Chapter 17. And as always, please make sure that you have completed your screencast study guide.